Hello, and welcome once again to another episode of Genetics Simplified, the podcast where we break down the most foundational discoveries that have shaped modern genetics. In today's episodes, we're going to delve a little further into the fascinating rediscovery of Gregor Mendel and his groundbreaking work on heredity. Although Mendel's work was published in and around 1866, it wasn't much later until like the 1900s that three scientists, Hugo de Vries, Carl Correns, and Erich von Tschermak, independently brought his findings back to light. Interestingly, de Vries and Correns are believed to have stumbled upon Mendel's laws before ever even reading his original papers. Meanwhile, Schmark, understandings of Mendel's work may have probably only solidified after he encountered the same things in the 19th century publications. So, anyways, I think I'm getting ahead of myself. Welcome and join me on another journey on exploring the rediscoveries cemented by Mendel, the father of genetics. And a lot of inheritance talk today. But first, let's start with Hugo de Vries. Hugo was a Dutch botanist, geneticist, and an evolutionary theorist. He was born in Haarlem, the Netherlands, and was a professor of botany at the University of Amsterdam. When he began his groundbreaking experiments with plants in the 1880s, what interested De Vries what's interesting about De Vries is that he initially didn't know about Gregor Mendel or any of his works. Obviously, De Vries would come to find out much later about the pea plants. But despite this, De Vries reached similar conclusions through his own hybridization experiments. And around the 1900s, he published his first findings. They were first published in French and then in German. But that's where things get a little more controversial because the French report didn't mention Mendel at all. And there was a 2-year gap between the French and the German publications. But by this time, his German paper had already come out and had acknowledged Mendel's contribution. So the big question back then, why the change? So some believe that De Vries might have come across Mendel's work after publishing his own and realizing that there were others who were also aware of Mendel. He added the reference, obviously, but even suggested that De Vries may have thought his conclusions were more advanced than Mendel's. But De Vries didn't just stop with rediscovering Mendel's laws. He had his own ideas, especially about how species evolve. So while many scientists of his time thought evolution happened gradually, De Vries proposed a very sudden and dramatic change, which is what he called as the discontinuous variation. He developed what became to be known as the mutation theory, and through his work with the evening primrose, the Latin for Onothera lamarckiana. Correct me if I'm wrong. I'm pretty sure I've got it wrong. But anyway, during his experiments, De Vries had noticed that these plants sometimes produced offspring with significant differences, especially the changes in leaf shape or plant size. And a lot of times when the new traits were passed down to future generations, De Vries classified them as entirely new species. So now we know that De Vries was partly right, but not for the reasons he thought. Because what he observed in the evening primrose wasn't really caused by a gene mutation, as he believed. Instead, it was due to chromosomal anomalies, essentially just errors in how chromosomes were divided during reproduction. So while De Vries may have misunderstood the full picture, his work was still important, quite incredibly actually. And his mutation theory definitely helped push forward our understanding of evolution and genetics, even if some of the details weren't quite right. And that's what makes science interesting. Anyways, let's slowly shift our focus onto the next. Carl Correns, a German botanist and geneticist, another very key figure in the rediscovery of Mendel's work. He was born in Munich, in Germany, and Correns faced a very challenging start to life. He lost both of his parents at a very young age. He was raised by his aunt in Switzerland, and he later pursued botany at the University of Munich in 1885. There he connects with Carl Nageli, a botanist who had corresponded with Mendel about some of his experiments, unbeknownst to Carl Correns at the time. But Nageli took a very personal and professional interest in Correns, often encouraging his passions for botany and guiding his academic work. Interestingly, this bond went beyond science. Correns goes on to eventually marry Nageli's grandniece. And in 1892, while working as a tutor at the University of Tübingen, Correns begins his own experiments on plant inheritance. Though he wasn't initially aware of Mendel's famous pea plant, the results thereof, it wasn't much later until the 1900s when Correns publishes his work, where he references Mendel's findings. And the paper itself is titled G. Mendel's Laws Concerning the Behavior of the Progeny of Racial Hybrids. Correns, along with De Vries, were instrumental in redefining Mendel's laws, clearly stating that we now know as Mendel's laws of segregation and law of independent assortment. Correns wasn't concerned with personal credit, though he even expressed frustration that De Vries had initially failed at mentioning Mendel in his first publication, which was in French. For Correns, the rediscovery of Mendel's laws was just a step towards furthering his own research, which I'm sure he considered more important. And coming around to, I think, the 1913s, Correns becomes the first director of the Kaiser Wilhelm Institute for Biology, which is in Berlin. And a very prestigious position, but unfortunately, a lot of Karl Correns' work is lost or destroyed during the bombings in 1945. Nevertheless, his contribution to genetics remained significant, a lot of which was somehow salvaged and saved, essentially giving Mendel the recognition he deserved. Now, the third scientist, the one who hadn't corroborated or worked with Correns or Nageli, or for that matter, De Vries, we come to Erich von Tschermak. His full name was Erich von Tschermak Senseneg, born in Vienna, Austria. Tschermak came from a very scientific family. His father was a mineralogist, and his maternal grandfather, Edward Fenzel, had even taught Gregor Mendel. Tschermak studied agriculture at the University of Vienna and later earns his doctorate from Halle-Wittenberg University. In 1898, he begins plant breeding experiment with peas, and by the 1900s, much like the other two scientists we mentioned, derives the same laws of inheritance as Mendel. Now, although he was younger and less established in the scientific community, Tschermak quickly publishes his results and earns recognition as one of the rediscoverers of Mendel's work. Tschermak's focus was practical. His hybridization experiments aimed to improve crops. He personally bred high-yield varieties of wheat, barley, and oats, making a significant impact on agriculture in Austria. And in 1903, he became an associate professor at the University of Agricultural Sciences in Vienna, later rising to a full professor, cementing his influence in plant breeding. But as news of their simultaneous rediscovery spread, the three botanists become aware of each other. And while each had made the discovery independently, their paths were now intertwined. Science thrives on collaboration and mutual reinforcement. And a little unfortunate, but quite fortunate at the same time, the rediscovery of Mendel's laws were no exception. But the interaction between De Vries, Correns, and Tschermak wasn't without its own complications. So the publication of their findings comes out in the 1900s, marked what I would call a watershed moment for genetics. Each of them publishes within months of each other, sparking intense debates with the scientific community on the tightrope about the origin of these insights. Initially, De Vries appeared hesitant to acknowledge Mendel's priority fully. He suggested that his own research had arrived at the same conclusion independently, but this didn't sit well with Correns, who was adamant that Mendel deserved the full credit. Correns was known for his integrity and his loyalty to the scientific accuracy. He wasn't shy about voicing his opinions. He wrote to De Vries, pointing out that Mendel's laws had been established decades before any of them even undertook their experiments. Now, this difference in attitude towards Mendel's legacy definitely created a little bit of a rift and some tension. So, De Vries had the international reputation and resources to promote his findings. Correns was driven by a very strong sense of scientific ethic, and he sought to ensure Mendel's work was respected. But Tschermak kept a lower profile, contributing to the rediscovery in his own way, but staying out of the rivalry between De Vries and Correns. Now, around the same time is an entirely new field of scientific reinforcement and rediscovery. In 1902, the first statistical analysis of Mendel's data is published. In the first volume of Biometrica, published in the book of Weldon in 1902. Now, using the then recently invented chi-square test, which is now referenced to a paper by Pearson from the 1900s, there rises an intersection between genetics and statistics, which in itself is a very fascinating tale that underscores the importance of statistical methods, especially in validating scientific discoveries. Now, this half of the story is going to revolve more around the development of the chi-square test, the Fisher-Mendel controversy, and the profound implication that this had on future statistical practices and research. Now, for those of you who have read the blog, I did leave a little bit of a Easter egg for you guys that this was going to come, that we would be discussing this. I couldn't help but not add it on this podcast. But I feel like this is as important as talking about genetics itself, because the applications of this go far and wide, not just for genetics, but for any form of scientific discovery. This is going to be relevant. So in the world of scientific research, a few statistical tools have been as impactful as the chi-square test. Now, a method for assessing the relationship between observed and expected data is what the chi-square test is going to be important for. And this tool becomes a staple in proving the significance of research findings, particularly in genetics and biology, especially in the context of the story. But the origins of the chi-square test are tangled with one of the most famous controversies in genetics. As I told you, the Fisher-Mendel controversy. Now, this dispute, fueled by questions over Gregor Mendel's data and Ronald Fisher's analysis of it, led to significant discussions about reliability of scientific findings and established standards for statistical integrity. In 1936, the renowned statistician and geneticist Ronald Fisher revisits Mendel's original data. Fisher was back then a very prominent figure in the development of statistical sciences and kept a very keen interest in applying statistical methods to biological problems. His paper, titled "Has Mendel's Work Been Rediscovered?", applied the chi-square test to Mendel's results and made a very startling observation. Fisher noticed that Mendel's data matched the expected ratios a little too closely, closer than what would typically be expected from a random sampling. Even the variations did not match. So in statistical terms, the probability of obtaining the results as close as Mendel's purely by chance was too low to be true. So this led Fisher to speculate that Mendel's data might have been influenced by an unconscious bias, or that some results were adjusted to better fit the expected ratio. Now, I would call that a scientific bias in order to publish, so that, hey, let me just change a couple of numbers here and see, maybe this I can publish because these are results that shouldn't be outside this margin. Could be, I'd be going a stretch too far to say maybe it was fabricated data, or maybe that was the implication. Regardless, the chi-square test was developed in the early 20th century, and as a method of testing the goodness of fit, a way to compare observed results with the expected frequencies under a very particular hypothesis. In its simplest form, the chi-square test allowed researchers to determine whether observed deviations from expected outcomes are due to chance or if they indicate a significant relationship. For example, if we expect a 1:1 ratio of the manifestation of two traits in a genetic experiment, we can use the chi-square test to determine if any deviations from this ratio are statistically significant or whether it is just a simple random variation. The chi-square test quickly gains popularity for its versatility in frequency testing, categorical data testing, and was particularly used in the fields such as genetics, where scientists could test hypotheses about inheritance patterns, Mendelian genetics especially. Anyway, Mendelian genetics, for that matter, which predicts specific ratios for different traits, provided fertile ground for applying this test. The application of the chi-square test to Mendel's work led to one of the most famous controversies, and it wasn't without a warrant, such as the 3:1 ratio for dominant and recessive traits in his F2 generation. For years, Mendel's work was accepted without question as it provided the foundation for modern genetics. But Now, in 1936, a British statistician and geneticist takes a closer look and says, it's too good to be true. He observed the data and noticed that it deviated less from what was expected. Maybe it was unconscious, or maybe it was done unconsciously, or maybe it was intentionally adjusted to fit the data better. Anyway, regardless of this, Fisher's analysis did suggest that the results were suspiciously close to the expected ratios, far more than typical biological data, which usually exhibits some natural variation. Now, this statistical anomaly led Fisher to hypothesize that Mendel's data might have been fudged or selectively reported. This claim cast a massive shadow over Mendel's work and raised fundamental questions about the reliability of his findings. Before I explain further about the relevance of why those three scientists gained their importance in Mendel's rediscovery, I'm now going to tell you about the Mendel controversy, which I think I have covered in a very basic gist. But those three scientists coming into the picture, independently proving Mendel's rediscovery with three separate plants, is probably the goldmine that gives Mendel credit. But I'm getting ahead of myself. The Fisher-Mendel controversy had far-reaching implications, and especially for the scientific community, sparking debates about data integrity, replication, and the need for rigorous statistical analysis and research. So while Fisher doesn't directly accuse Mendel of intentional fraud, his findings led to a re-evaluation of how data should be presented and analyzed. Fisher's use of the chi-square test to question Mendel's results underscored the importance of statistical methods in evaluating the credibility of scientific data. So from this point forward, the chi-square test and other statistical tools essentially become vital for assessing the reliability of experimental findings. Researchers recognized that there is a need to report results transparently and to use statistical tests to verify the significance of their findings. But this controversy also highlights one other thing, the concept of statistical significance and the role it plays in determining the validity of research. So before the chi-square test and similar methods were commonly used, it was easy for scientists to report data that seemed to confirm their hypothesis without thorough statistical validation. Fisher's analysis set a standard for future research. He said data should not only fit a hypothesis, but should also pass the test of statistical scrutiny to rule out the possibility of bias or chance. Now, the chi-square test remains that fundamental statistical tool in research today. Its application extends beyond genetics to fields like psychology, ecology, epidemiology, market research, so much more. The test is essentially useful for three main things: the goodness of fit testing, the test of independence, and the homogeneity testing. Now, by providing a quantitative measure of the discrepancy observed between the observed and the expected frequencies, the chi-square test enables researchers to make informed decisions about the validity of their hypothesis. And the story of the chi-square test, along with the Fisher-Mendel controversy, illustrates the importance of statistical rigor in science. It has broad implications for how we interpret research findings today. The controversy in itself catalyzed a shift towards more rigorous standards in scientific research. It made clear that statistical significance should be an essential part of scientific inquiry, especially in fields where experimental results might vary due to natural biological variability. Now, I need to go through my notes for this because this is too much even for me to have to find a way to remember. The shift that the implications of the Mendel-Fisher controversy gave rise to three or four main things. Actually, I would go a stretch more to say five. I'd say one of them is the reproducibility and transparency. It underscores the importance of transparency and reproducibility in research for the simple reason that scientists begin to publish not only their findings but also their methods and raw data, which enables others to reproduce and validate their results. The concept of the p-value and thresholds for statistical significance became standardized as a way to quantify the reliability of findings. The rigor of statistical testing. The chi-square test and other statistical tests became widely adopted to determine likelihood that observed results were due to chance. The chi-square test specifically allowed researchers to assess categorical data and test hypotheses involved expecting frequencies, which was particularly useful, especially in fields such as genetics, medicine, and social sciences. Now, Fisher's analysis brought attention to the possibility of an unconscious bias in data collection and reporting. Now, researchers became more cautious after this, especially about ensuring objectivity in their work. Elements such as random sampling and sampling errors and biasing errors are hindrances in the objectivity. So to have a single blind or a double blind experiment, which you must pursue, you must read through some statistics to understand if you're in a scientific venture. But the awareness eventually led to the development of various other experimental controls to minimize these biases. And they are both very, very important. The last two being the ethics in data reporting and the interdisciplinary applications. The controversy prompted scientists to establish ethical guidelines for data reporting to prevent researchers from publishing and fabricating evidence and data that would not make sense. It was hard to determine what is possible from what is not, and it was essential to be able to replicate. Today, researchers are encouraged to report all data, even if it doesn't fit neatly with expected outcomes, as this honesty is essential for the integrity of scientific knowledge. And the chi-square test's utility is what proves it, because it quickly spread beyond genetics to other fields, from epidemiology to psychology, where researchers could test relationships between categorical variables. It becomes an indispensable tool in public health research, allowing scientists to study associations between diseases and risk factors. But this only brings us to the end of today's episode of Genetics Simplified. Now, we have explored a little bit more about De Vries, Carl Correns, and Erich von Tschermak, not to forget the perks and I cannot English anymore. The tool of chi-square test and statistical significance that gets brought into the picture. But in a world that is now changing faster than ever with the advent of the 20th century, discoveries are anew, inventions are rampant and growing faster. Industrialization is slowly picking up more and more steam, and in a world that is slowly working on more lethal weapons, there's a world of scientists who are also finding cures and understanding the very fabric of what makes us us. So thank you so much for joining me on this podcast. My camera just overheated and died. So I was prepared with a backup camera. But thank you so much. And please leave me a thought, a note, anything you may have as a comment, anything you want answered, any questions. I'm always happy to engage the viewers that I have and grow it more slowly and steadily. But if you have anything you'd like to discuss more on this, please leave me a comment. Don't forget to like, subscribe, share, and see you once again, hopefully on another episode before the Christmas. So goodbye and take care, and thank you so much.